Hi, I'm Camille Marchet. Today I will be presenting REINDEER, the data structures to index large collections of datasets, raw reads in particular. So let's start with some numbers. Here you have a figure coming from SRA with the current number of bases that are stored on SRA servers. So at the moment, millions of billions of bases. And while it's, downloadable, available, it's very hard to use this data as is on the servers. For instance, if you have a query of interest, you cannot know where this sequence in each of the datasets it appears. So in this talk, I will be talking about growing literature of data structures that allow to index and query sequences in large collection of such datasets. And I will present REINDEER and explain why it's different from the rest of the data structure and show some results on eukaryotes. So with this growing literature of methods that allow to index large collections of raw reads, we saw some new applications. For instance, people tried to look for fusion events in 10,000 RNA-seq data coming from cancer datasets. Also, applications to microbes with looking for resistance genes in collections of microbial datasets, or more recently, these kind of structures were used to index different genomes and to help building phylogenetic trees. So what are these methods doing exactly? They see a set of datasets like this one as a collection, and they imply that you have a query sequence that you want to look for in each of the datasets, and you want to return all the datasets that contain this query. And in fact, you can have, of course, several queries, and in this case, for each queries, you want to return all the datasets that contain the query. So in 2016, Solomon and Kingsford proposed a solution for this problem by indexing, instead of the raw reads, indexing the k-mers of each datasets. So you see each dataset as a set of k-mers, and it's the same for the query. You convert each sequence to a set of k-mer, and then you look for an intersection between the query and each dataset. And you want to report the dataset that have a sufficient intersection with the query. And by sufficient, I mean some parameter. So for instance, if my threshold for the intersection is 4, I would report this dataset that contains 5 k-mers from the query, and also this one, in, for instance, a vector of booleans, saying, yes, my query approximately is present in the first dataset and in the third one. So what we bring here with REINDEER is to be able to query abundances. Indeed, with the previous method, you could only say if the query is present or absent. But you would like also maybe to access the abundance of the query. So you can count k-mers, and in this case, you index multi-set of k-mers, in fact. And let's say that for the k-mers of the query in dataset 1, each k-mer has a count value of 16, for instance. You would like to return 16. So here in the vector, that it becomes a vector of integers and different values for each of the datasets. So this is what we do in REINDEER. But first, a bit of literature about what was existing before. So it started in 2016 for the large-scale data structures. And the literature, we can divide it in two ways of doing. So if you want to know a bit more about all these methods, you can refer to a survey that we did recently that is available on bioRxiv. And just a few words. So the first type of methods are k-mer aggregative methods, meaning that for each of the data sets, they represent the set of k-mer independently. And then they organize all those sets in another structure that allows to do quick queries, such as, for instance, a tree. And the other way is to take the union set of all k-mers from all the data sets, and for each of them, having a corresponding presence and absence vector. So we call that a color matrix. And REINDEER is closer to this second way of doing. But with none of these methods, you could access the abundance of a given query. So why would you like to access the abundance of a query? For instance, if you have a mutation in a bacterial strain that you would like to monitor across different data sets, well, the idea would be that not only you want to see if it's present or not, but you maybe want to see if it's very frequent or quite rare, because it has impacts on the biological conclusion you will make. So this is just an example, and there are other applications of, such as, for instance, RNA-seq, that is very straightforward. So what we do in REINDEER, a very general way of thinking of it is just thinking of a matrix. So I said that for some methods, you would associate to each k-mer its presence or absence in a Boolean vector in each of the datasets. And for REINDEER, what we do is associating to each k-mer a vector of integers with its abundance in each of the datasets. So a prerequisite is to count each k-mer in all the datasets. So you can do this with many tools such as KMC. And then we need three things. First, record this set of k-mers to know which k-mers were here in the first place, were in one of or several of the data sets. Associate each k-mer to a count vector like this, and store these integer vectors. So we focused on three components in the index. First, recording this set of k-mers and trying to do it efficiently. Second, associating the k-mers to their counts. So we did this with a hash table that is specialized for k-mers. That is a previous work of my collaborators and mine, and on focusing on the counts that are stored in the matrix. So first, about indexing the k-mer set. This relates to recent literature about spectrum-preserving string sets. So spectrum-preserving string sets are string sets that represent exactly the information in a set of k-mer. And you can easily see that there are different ways to represent the information of a set of k-mer. So let's start with this example. You have here two data sets, D1 and D2, and here you have the union of the two data sets. Well, a way to represent the information of the k-mer set is to record independently each of the k-mer, for instance. But you can also, doing assembly, build a unitig for all those k-mers. And the difference is that you use less bases, because you took the first k-mer and then just added the last nucleotides of the following. So it's more compact. And you have different other ways to represent a k-mer set more and more efficiently, but none of these SPSS would ensure for us to record a single and non-ambiguous count vector per each k-mer. So we came up with another solution, which is monotigs. So again, let's start with these two datasets, D1 and D2. We have this full set of the union set of k-mers from D1 and D2. Some k-mers have a count in D1 but not in D2, or a count in D2 but not in D1, or a count in D1 and D2. And let's say that these two counts here are the same than here, here, and here. So the intuition behind monotics is to perform assembly if and only if the k-mers have the same count vector. So here we would start with this k-mer, see that we could assemble it because it has the k-1 overlap with the second one, but the count vector is not the same, so we don't assemble. Then we start again with the second k-mer and see that we can assemble it with 3/1s. So we end up with this sequence, the second one, and then the third sequence. So here you see that we use assembly not to build a doubling graph in the sense that we want to assemble the full information of the k-mer sets. We use assembly as a way to represent more compactly k-mer set. In practice, the k-mers are greedily assembled in partitions so that we can scale to the billions of k-mers we have to handle. All right, so to sum up, we have all those k-mers. We compute the monotics for the sets of the union set of k-mers. So remember that here, having these dots here means that you have the same counts in the data sets. And then you can associate for each monotic one row in the matrix. So why? Well, because we have this nice property that a k-mer is found in one and only one monotic. So each monotic can be associated to a non-ambiguous vector in the matrix. Finally, we worked on shrinking the matrix. So two ways to do this. First, noticing that we can possibly have identical rows in the matrix. Why? Because of the way we build the monotics. Two monotics can have the same count vector. So the first thing we did was to deduplicate the rows in the matrix so that we could reduce the row number in the matrix. Second thing is to notice that the matrix can be quite sparse. So we used run length encoding compressing methods to reduce the row length. And each time we want to access a row to know the count vector of a k-mer, we can decompress this row and have all the counts. So REINDEER is available on GitHub. The input at the moment would be a unitic file from Bekarm so that we have all the k-mers and their counts readily available. The query would be for each of the query sequence, one line with the header of the sequence and then all the counts in each of the datasets. And something that is nice with REINDEER is that if you have built a REINDEER index on a large collection of datasets, you can dump the index on the disk and then reload it to do new queries. So let me show you some results on the eukaryotic data. So we indexed 2,500 human RNA-seq that were previously used in several benchmarks of the literature. We shown REINDEER compared to those two ways of doing in the literature to index presence and absence, so different methods here. Also for the record, we included a version that only stores presence and absence in REINDEER, and the regular REINDEER with the counts. So this is the only one that can handle count information. And because of this, we do not expect REINDEER to have the best performance results because it has more information to record. So let's start with the time. You can see that it's It's between 2 and 55 hours to record such a large collection. REINDEER is 45. About the memory, the memory, we are at 56 gigas. And for the index size, we end up with 52 gigas, which is not the biggest index, in fact, if you look at the column. And you can notice too that this final index size is only 1% of the size of the raw data, which was around 5 terabytes. Also, for the query, we queried 10,000 transcripts and shown that each query was under the second. And if you want to look at the paper, we have an additional experiment looking for oncogene sequences in this collection of RNA-seq. So a recent application of REINDEER was to index COVID data. So data sets coming from patients, mainly amplicons, containing viral genomes. So this was done at Pasteur Institute. You can go and check at the URL and try to do a query. So if you know a mutation of the COVID that you would like to see, if it's spread across a population or across, after a date, you can look at it and have not only the presence of the mutation, but also the abundance in each of the datasets. All right, this is the end of my presentation. So to sum up, we deal with a lot of data sets at the moment, and we have trouble indexing them at a large scale. Thanks to some new methods, we are able to find presence and absence of query sequence of arbitrary length in collections of data sets. So mostly we are being able to index a few thousands of eukaryotic data sets with those methods. And REINDEER brings the contribution to be able to query the abundance of sequences in these kind of collections. So since it's alignment free, because it's k-mer based and we are also based on hashing, we perform very quick queries on those data sets. And we also introduce a new k-mer set representation using strings called monotics. You can find our work on GitHub, and it was accepted in ISMB. You can find also the article currently on bioRxiv and soon on bioinformatics. Thank you to the organizers and to my co-authors and people who helped me with this work. And now I will take your questions.